beloved students. Today we are starting with a new chapter, genetics. But before starting this chapter, I have an announcement for you. And an announcement for you is that we have UCMLE notes and MBBS notes along with question banks are available. Use it and score it, high ranks and medical journey. We have collected UCMLE notes all around the world. These notes are prepared by the top faculty of the teachers who prepare students for UCMLE. So we have those notes available as well as MBBS notes available along with lecture series. So if you will use these notes for UCMLE, I think so you will score higher. And if you are an MBBS student and you want to use these notes for MBBS, I think so you will score higher. I promise you will score higher. If you are interested to avail these notes, contact me in my personal WhatsApp number. I will provide all the samples of these notes, and you will see how these notes are prepared, especially these UCMLE notes. These are prepared in 4 different colors, means both MBBS and UCMLE, 4 different colors. One color for fast MCQ, one for future, and one for important clinical. Like that way these notes are prepared. Just check it and then decide whether to avail or not avail. But these notes are very important for you. I mean, you will enjoy. And especially the question bank with UCMLE like FastEd, Kaplan, Emboss, all the question banks are also available. So you can use it and score it. If you want to interest it, I will mention my personal contact number in the comment box and contact me there. I will full support you, especially for UCMLE. I will full support you. Guidance, if you need guidance for UCMLE, I will support you, because being teaching UCMLE for many years, I have vast experience regarding UCMLE. So contact me and I will guide you, and I will try my best to guide you in the right path where you can achieve your goals, where you will score higher with dignity. So let's start the chapter. So let's start this chapter, genetic disorders. Genetics basically deals with the study of the genes, and the genes are divided into 2 parts. One is called coding gene, one are non-coding genes. What is meant by coding genes? Those genes which prepare protein, and non-coding genes mean regulatory genes which regulates the protein. Regulation is done by non-coding genes. Coding genes are also called exons, and you can remember that exons express the formation of proteins, and the non-coding genes are called intron. If we consider 100% of genes, exon constitute about 1.5% of the genes. That small much are genes express protein, while 98.5% consist of non-coding genes. Then non-coding genes are divided into 4 types. One is telomeres and centromeres, one is non-coding RNA, and one is jumping genes. So they are divided into 3 types: telomeres and centromeres, non-coding RNA, and jumping genes. Remember that jumping genes are also called transposons, and this is a very important MCQ question. They are also called transposons. Non-coding RNA include microRNA, long non-coding RNA, piRNA. And this piRNA is most common non-coding RNA in human genome. Jumping genes and transposons. Why it is called transposon? Because they continuously moves within our human genome. So therefore it is called transposon. And it constitutes 33.3% of human genome in 98.5% of human genome. And its function is chromatin organization. Its function is chromatin organization. So let's first discuss this one, microRNA. So microRNA constitute 5% of human genome. Got it? And its function is silencing RNA, gene silencing RNA. Gene silencing RNA. Post-transcriptional gene silencing. Gene silence means to stop. So gene silence means to stop the formation of protein synthesis. How it does what? Post-transcriptional gene silencing. That once transcription is done, after that it silence the gene and avoid the gene for translation. Two processes in the protein synthesis, one is transcription, one is translation. So it avoids translation. For example, this is a strand, this one is 5 region and this one is a 3 region. So this one is, for example, untranslated region, means its translation is not done. What will do? Here microRNA will attaches and make it untranslated region. For example, this is a strand, and if this gene want to silence this area, microRNA will attach here and will silence this gene. When this gene is silent, now it cannot do translation, so it will be remain untranslated. So therefore it is called untranslated region. And the microRNA, it consists of 21 to 30 oligonucleotides. So therefore they are also called microRNA because a small amount of oligonucleotides, 21 to 30. So its function is gene silencing. So if we use in a practical purpose, for example, this is microRNA, and microRNA if inhibits this gene, for example, there is a gene called tumor gene in our body. If this gene expressed, there will be formation of tumor. But if microRNA attaches to this tumor gene, there will be no formation of the tumor. And for example, there is a gene called tumor suppressor gene, means it suppresses the tumor. And if this microRNA inhibits this tumor suppressor gene, so there will be formation of the tumor. Now exogenous means artificial microRNA is prepared, and that RNA is called small interfering RNA. Small interfering RNA. So by genetic engineering, this small interfering RNA is prepared. Now genetic engineering having 3 technologies. One is gene knocked in, knocked out, and knocked down. What is meant? Knocked in means then when you add a strand, when you add DNA part to the strand. Gene knockout means when you remove a segment of DNA from a strand. And gene knockdown means then when you entered microRNA or small interfering RNA. So microRNA or small interfering RNA function is gene silencing. So this process is called gene knocked down. I hope I made myself clear. Now long non-coding RNA. Why it is called long non-coding RNA? Why it is called long? Because it is made up of 200 to 500 nucleotides. And its function is gene silencing and activation both. Gene silencing, for example, bar body formation. So due to inactivation of the ZIST gene. When ZIST gene is inactivated, we will describe this in future lectures. What is bar body formation? For example, why bar body formation take place? Because this gene always inhibits the formation of bar body. When this gene is inactivated, now bar body will be formed. What is meant by gene activation? For example, stabilize tertiary structure of protein by regulatory chromatin organization and gene activity. Gene is activated to stabilize the tertiary structure of protein. Got it? So it having both function, gene silencing and gene activation. Now genetic disorders, they are divided into three types: single gene disorders, multiple gene disorders, and chromosomal disorders. Single gene disorder are divided into two types: classical inheritance and unclassical inheritance. Classical inheritance means Mendelian inheritance. We have discussed in the lower grade autosomal recessive, autosomal dominants. They are included in Mendelian inheritance known as classical inheritance. Autosomal recessive most commonly in leads to inborn error of metabolism. Always, if you don't know the right answer and the examiner asks about the metabolism, that whether it is autosomal recessive or autosomal dominant, so mostly 90% the answer lies in autosomal recessive. Autosomal dominant, the most common pattern is this one. The most common single gene disorders are autosomal dominant, and autosomal dominant most common pattern in 65% of cases, and the other two are X-linked recessive and X-linked dominant. While in unclassic inheritance, we will discuss each and every one in detail. Unclassic inheritance include trinucleotide repeat mutation. We will discuss Huntington disease here. Mitochondrial inheritance, we will discuss it in a while. Genomic imprinting and gonadal mosaicism. While multiple gene disorder means that multifunctional inheritance or complex polygenic inheritance, it means that it involves more than two genes affected plus environmental factors, and these examples are diabetes mellitus, cleft lip, cleft palate, congenital heart disease, hypertension, coronal heart disease, and gout. While the chromosomal disorders means that there is a change in the number of chromosomes, numerical, or it can be a structural, means that there is a change in the number of chromosome or shape of chromosome. Numerical include euploidy and aneuploidy. Euploidy means exact multiples, means 23, 46, 69, and aneuploidy means that not exact multiple, means that 46 then 47 or like that. And structural disorders include deletion, translocation, ring chromosome, isochromosome, and inversion. Got this point? So these are about genetic disorders. Now let's discuss one gene. One gene is formed or imprinted from both mother and father. Means one gene is made up of two alleles, one from maternal and one from paternal. Got this point? Keep this in mind so we will easily describe genetic disorders. Let's start with autosomal dominant. Autosomal dominant included in classical inheritance, which is called Mendelian inheritance, and autosomal dominant is most common pattern in 65% of cases. Means this disorder is most commonly occurred, and this disorder is most commonly in metabolism diseases. If there is a metabolic disorder, so most commonly it is autosomal recessive. So let's start autosomal dominant. If autosomal dominant is heterozygous, autosomal dominant is heterozygous means one gene is one allele is affected, you can say, or one gene affected, so it is heterozygous. If both the genes are affected, means homozygous. So autosomal dominant means that one affected, it will carry the disease. If two affected, so the disease frequency will be high. While autosomal recessive means that when one affected, it means it is a carrier. When both affected, it means it is a disease. So let's start with autosomal dominant. In heterozygous condition, it is considered as disease, while homozygous, they always die in the intrauterine life. Got these points? So then in autosomal recessive, we have discussed that heterozygous will be considered as a carrier, while homozygous will be declared as a disease. So first we will discuss autosomal dominant. So autosomal dominant disorders, the first feature is variable expressivity. Means when single gene with different expression in different individual, it is called variable expressivity. Variable expressivity means that, for example, there is a neurofibrochromatosis 1 gene. There are three people, one name is Aslam, one is Chauhan, and one is Akbar. When in Aslam this gene is affected, this gene leads to a disease of macule. When in Chauhan it is affected, it leads to iris. When in Akbar it is affected, it leads to spine problem. So there is a same gene affected in different people, same gene affected in different people, but their expressivity is different, means that they express a different problem. So this is called variable expressivity. The second one is pleiotropism. Pleiotropism means that now in one body, when one gene affect and it cause different diseases. We have studied here, when in different individual the same gene is affected, it causes different problems. Now when one individual, the same gene is affected and it causes different diseases, it is called pleiotropism. So for example, there is a sickle cell anemia gene. When it mutates, it can lead to hemolytic anemia in the same person, and it caused lung problem in the same person, spleen problem, and bone problem. Now one gene causing different problems, but the individual is the same, it is called pleiotropism. I hope I made myself clear. The third one is incomplete penetrance. Incomplete penetrance, presence of mutant gene, but phenotypically it is normal. Presence of mutant gene, but phenotypically normal. That there is a presence of the mutant gene means that genetically the person is abnormal, but when you see the person is normal, it is called incomplete penetrance, because we have already declared that in autosomal dominant, if one gene is affected, the person will be declared as a disease. But there is an exception, that there is a mutant gene present, but he is looking normal. Let's consider an example, retinoblastoma gene present on chromosome number 13, and Q stands for long arm while P stands for short arm. 13 long arm, 14, this is the sub-region on the chromosome. So this gene is autosomal dominant. but this gene is normal. Why? How it becomes normal? So in heterozygous state, the man is in heterozygous state because we have already declared when there is a single gene affected in autosomal dominant, the person will be declared as a disease. But here it is normal. What happens? So there is a famous hypothesis present by Knudsen, which is called Knudsen two-head hypothesis. When this person born at the time of birth, one gene was from mother side and one gene was from father side. This was a mutant gene and this was a complete normal gene. But after some time, this gene also becomes mutant. This gene also become mutant. The second gene also become mutant. So now there is no heterozygosity because he was a higher heterozygous and he was normal. Now there is no heterozygosity because later on it acquires another mutation. So therefore the person looks normal. The fifth feature of autosomal dominant is dominant negative effect. Because when mutant gene is affecting the normal gene and products, it is called dominant negative effect. And it is seen in those genes encoding multimeric proteins like fibrillin and collagen. Fibrillin and collagen. Triple helix, for example, this is collagen. It is a triple helix, glycine, proline, and xanthine. For example, it is made up of 3 genes. For example, this glycine is mutant while the rest 2 are normal. When this mutant gene will affect these 2 normal genes, it is called dominant negative effect. So we will discuss the collagen diseases and fibrillin diseases now. Fibrillin mutation, it leads to a syndrome called Marfan syndrome, while collagen mutation, it leads to Ehlers-Danlos syndrome. Remember these 2 diseases and osteogenesis imperfecta because the 3 diseases are very common diseases and mostly asked in the questions. Fibrillin mutation leads to Marfan syndrome, collagen mutation leads to Ehlers-Danlos syndrome and osteogenesis imperfecta. And this effect is dominant negative effect. First of all, discuss Ehlers-Danlos syndrome. What is Ehlers-Danlos syndrome? So, heterozygous disorder consisting of Ehlers-Danlos syndrome having 7 types, type 1 to 7, and because of multiple collagens are affected. So overall, autosomal dominant is most common pattern and collagen 3 is most commonly affected in Ehlers-Danlos syndrome. Type 3 is most common while type 7 is least common. How will Ehlers-Danlos syndrome patient present? So these are the clinical features like hyperextensibility of joint plus skin, rubber-like density, rubber-like elasticity because there is hyperextensibility in the joints and skin. So this man will be looking like a rubber man. So therefore it is also known as rubber man syndrome. While abnormal scar tissue, thin cigarette paper like scarring will be present. Abnormal scar tissue will be present on the body, and there may be a spontaneous rupture of colon plus arteries. So Ehlers-Danlos syndrome type 4. Spontaneous rupture mostly occurs in Ehlers-Danlos syndrome type 4. It is also known as vascular Ehlers-Danlos syndrome and it having worst prognosis because it leads to sudden death. The next is osteogenesis imperfecta. It is mostly common in pediatric. There will be a fracture of the bone, but this is a most common. Blue sclera. When you will see a blue sclera of a child, it means that he is suffering from osteogenesis imperfecta. Here is type 1 collagen affected, and here collagen type 3. The last one is Marfan syndrome. Autosomal dominant chromosomal. This disease is on chromosome 15 long arm, and here is a fibrillin 1 gene mutation. Fibrillin 1 gene mutation, it can also be due to tumor growth factor beta. So fibrillin function is to stabilize the three-dimensional structure of fibrillin protein, but when the and elastin contain tissue affected. Stabilize and fibrillin stabilize which protein? Elastin. So where elastin is present, because elastin, when fibrillin mutation occurs, it will not stabilize the elastin. So where elastin is present, there will be a defect occur. For example, in aorta, in the lens, in the heart. So the clinical features are triad. 3. You will look towards skeletal system for eye and for heart, cardiac system. And skeletal deformity is the most common characteristic. He will be a tall stature, thin and long fingers, also called archanodactyly, sorry, a spider like thin and long fingers, hyperextensibility of the joints, while eye, lens dislocation and retinal detachment. While in the cardiac problem, cystic medial necrosis, aneurysm, regurgitation of aorta, you can say mitral valve prolapse, and due to heart, it is the most common cause of that dissection of aorta in the Marfan syndrome. So diagnosis is done by revised giants criteria. Revised giant criteria say you the person suffering from Marfan syndrome must have a family history. He must have a fibrillin 1 gene mutation and characteristic signs and symptoms. So that was all about the chapter topic or lecture first of genetic disorders. So once again, I have an announcement that if you wanted to score higher ranks in medical journey, so and or if you are interested in UCML especially notes or MBBS notes along with question banks are available, you can score high, very high score. I promise it. I will guide you till your exams, and especially if you are interested in the UCML, I will guide from the core of my heart. You all are welcome. You can purchase or you can avail all these notes, and I will guide you how to use these notes, because we have collected these notes from the top faculty of the world, and you will be the topper if you use these, because it will save your time, it will potentiate your concepts, it will make you a star of the class. Keep study, and have a good time. Goodbye.